Hello, everyone. I'm Bhavna, a visiting faculty at Data Science Department, ISR Bhopal. I will be presenting our recent work on a deep learning framework for quantitative analysis of actin microridges. So let's get started with what are microridges. To elucidate this, I am showing a cartoon of the zebrafish epidermis around 48 HPF, which is bilayered, consisting of the basal epidermis at the bottom layer and periderm as the top layer. The apical surface of peridermal cells are modified into actin-based protrusions called microridges. These are evolutionarily conserved protrusions found on various non-cornified squamous epithelia. They form laterally long labyrinth patterns on peridermal cell surfaces as seen in the SCM image of the zebrafish embryos. The traverse section shows the actin projections in the TEM images. Recently, a number of actin regulators have been found to localize within the microridges. Importantly, the function of the ARP2-3 complex is essential for their formation and maintenance. We have been interested in having a quantitative description of the dynamics of microridges that required high-quality image data followed by appropriate analysis tools. In order to achieve this aim, we built a setup for live imaging of the zebrafish periderm cells. The embryo dimensions were measured during 2 to 2.5 days post-fertilization developmental stages. We then custom designed a mounting device for lateral imaging the epidermis using an upright microscope with a 40x lens objective. The head, yolk, and flank regions of the embryo were imaged as indicated by these red boxes. A eutrophin GFP probe faithfully captured the actin dynamics of microridges as seen in this movie on the right. With this setup, we wanted to build a framework to infer microridge dynamics using quantitative image analysis. I will walk you through the custom-built image processing pipeline step by step. Firstly, we made 3D time-lapse movies by live imaging the epidermis from apical to basal in the Z direction. The image entropy-based filtering steps eliminated the basal epidermis. The mean intensity of periderm cells patterned with microridges was stored as 2D time-lapse image sequences. In step 4, Gaussian smoothing diffused the microridges and enabled cell membrane segmentation. To extract single cells, we multiplied the Gaussian smooth image with the bounding box coordinate of each cell. To precisely extract polygonal shaped cells as they are, each cell was cropped with its bounding box and multiplied with its convex image. Single cells were tracked to be able to follow their microridge dynamics. Finally, custom codes were written to achieve microridge segmentation at single pixel level, consisting of two parameters, one for the level of Gaussian smoothing and the other for emphasizing the gradients. Using this image processing pipeline, we automatically extracted pairs of single-cell grayscale images patterned with microridges and their corresponding labeled images for CNN training. Our method alleviated labeler fatigue and increased throughput for CNN training. The idea here is once trained on such a set, when the trained network sees a similar image, it can automatically find the microridge labels. We implemented data normalization and image data augmentation by means of translation and rotation. The training data was partitioned into 90, 93, and 95% as training set and remaining as test set respectively, and these were passed through a UNet segmentation network. The output performance were assessed by using Jacquard similarity between the labeled versus the network predicted pixel level segmentation. After rigorously testing for various combinations of hyperparameters, we achieved 95% pixel-level segmentation accuracy. Here is an example showing pixel-wise segmentation of microridges within a yolk cell. Next, we tested the trained network performance on unseen data acquired with different signal-to-noise ratio than the training set. Although not trained on this data, the trained CNN produces reasonable segmentation results. On the other hand, the microridge conventional algorithm with default parameters resulted in severe oversegmentation. And only after fine-tuning the parameters, better segmentation result was achieved. Together, our results indicated that CNNs are better adaptable to produce robust segmentation. In order to test the CNNs a step further, we produced artificially contrast-altered images, high contrast and low contrast for each image within the training set. The conventional algorithm and its parameters were fine-tuned for each contrast-altered image type separately. Since the previous conventional algorithm parameters produced severe pixel-level under-segmentation. A single trained network on contrast-altered dataset with two different types of signal-to-noise ratio images produced about 84% accuracy. Our tests conclusively indicated that trained CNN models are versatile, can serve as high-end feature extractors, and even accessible to non-experts. Our CNN strategy consequently improved data quality required for in-depth quantitative analysis. The parameters extracted here required a large number of cells and thousands of microridges, which was only possible because of the quantitative methods developed in this work. We observed differences in the pattern parameters between different regions of the same embryo, such as pattern wavelength. The estimated persistence length of microridges, or the characteristic length at which a microridge bends to assess their inherent mechanical stiffness, found to be about 6.1 micrometers. From the single extracted and tracked cell time-lapse movies, we performed flow analysis. Strain field analysis revealed the presence of both tensile and compressive strains within the microridges. Furthermore, positional fluctuations of high-intensity actin clusters influenced pattern rearrangements over short length and time scales. In conclusion, the methods developed here can be adapted to other studies using quantitative imaging. Our large-scale quantitative analysis revealed some of the key aspects influencing microridge pattern dynamics. All the details can be found in our recent publication. And thank you very much for listening.